All right, so our last macromolecule are nucleic acids. We're going to go through this a little bit. We're not going to spend a whole lot of time on nucleic acids because we have a whole unit on them later. But I just want to give you the background as our fourth macromolecule. All right, so we've talked about functions. We have energy storage, all right, with carbohydrates and lipids and insulation. And remember, proteins have a wide variety, but basically nucleic acid is to pass along information and to store that information in the nucleus to be able to be passed on to the next generation. All right, so nucleic acids are different than a lot of the other ones we've talked about because there's really only two examples, and that is DNA and RNA. The N and the A in in the DNA and RNA stand for nucleic acids. All right, so we'll get to that here in a little bit. All right, so let's take a look at nucleic acids. So obviously, like I said, we know that the DNA is our genetic material. All right, it stores information in genes. Genes are basically just segments of DNA. All right, and those genes are blueprints for telling us how to build proteins. So we just did proteins. Proteins are made up of amino acids. Well, there is a code that's in the DNA that tells us how to put together those polypeptides. What order of amino acids makes those polypeptides, right? So that blueprint is in DNA. And just very briefly, we go from DNA to RNA to proteins, right? So those proteins are what gives us our traits. So if we can make insulin, everything works fine. If we don't, we're diabetic, right? So if we make melanin, a certain type of melanin, we have darker skin. If we make a lighter type of melanin, we have lighter skin, all right, and so on and so forth. So a lot of proteins lead to our traits. All right, so those segments of DNA, each segment, each gene tells us how to build a protein. Which amino acids? What's the primary structure of that polypeptide? What's that amino acid sequence in order to make that, right? That's what DNA does. It directs how to build that. All right, so obviously then we have to be able to transfer that information. We know that our DNA also helps us build new cells and gives us our traits and our instructions for the next generation, right? It gets passed on. So it has the ability to be copied and then passed on to the next generation. All right, so DNA, obviously, remember I said proteins are probably one of the most important, probably this is more important because DNA directs how to build proteins. So I would say these are the most important macromolecule, but it's not like we eat to get nucleic acids in our food, right? It's just something we have and we have to build ourselves. All right, so that's the function of nucleic acids. So what are they made of? So this is just showing you, like, this is a chromosome. It's made up of DNA, and like this is a gene, which is a segment of DNA that codes for a trait. So it codes for us to build a protein. So this is what DNA looks like unwound. You've seen the double helix before. So basically this is that small section of a gene sort of magnified. All right, that's all that is. All right, so what's DNA made of? What's its structure? As I said, there are only two examples. There's RNA, which is ribonucleic acid, RNA, and then there's DNA, nucleic acid. All right, not this. All right, DNA. All right, is the second nucleic acid, and just some real simple differences. When we do the unit, we're going to learn more. But real simple differences, RNA is only a single helix, all right, a single-sided strand nucleic acid, and DNA is double-sided, right? So this is the double helix. You'll get two sides bonded to each other, whereas RNA is only ever one-sided, all right? And the monomers of nucleic acids are called nucleotides. So this is a polymer. We do put together a whole bunch of nucleotides to make a nucleic acid. All right, so remember, lipid is our only non-polymer of these macromolecules that we've been talking about. Okay, so what's a nucleotide look like? A nucleotide consists of 3 parts. It's got a nitrogen base, a sugar, and a phosphate group. So just here is an example. Here is the nitrogen base. The nitrogen base is either one sort of ring shape here. Notice over here, so this is a nucleotide, and this nitrogen base has 2 rings. So there are some bases, I'm going to show you that here in a second, some bases have 1 ring, all right, and some have 2. But this yellow part is the nitrogen base. It's mainly made of carbons and nitrogens, okay? That's why it's called a nitrogen base. There is a sugar. It is a 5-carbon sugar, so those monosaccharides were 6-carbon sugars. This is a 5-carbon sugar. And here's what makes the difference between RNA and DNA. The sugar in RNA is ribose, therefore ribonucleic acid. The sugar in DNA is deoxyribose, therefore deoxyribonucleic acid. So it's missing an oxygen, which is why it's deoxyribonucleic acid. And the pentose sugar looks like this. All right, it's this green part here in a nucleotide. All right, so we've got the pentose sugar hooked to the nitrogen base. And then the last part, oh, here's the difference. Here's deoxyribose, here's ribose. So you can see they're very similar, except for right here is ribose. It has an OH, and here has an H in deoxyribose. That's really the only difference. And then the last part, the third part of a nucleotide, is a phosphate group, which is here. And you can see that phosphate group attaches to the sugar, and the sugar is in between the base and the phosphate group. So you can see how we just hook together through dehydration synthesis. We hook together a phosphate, a sugar, a base, all right, and then that sugar hooks to another phosphate sugar base, all right, so this makes up, all right, our polymer, right? We just keep hooking together a nucleotide, another nucleotide, another nucleotide. So this is showing you one side, and then if you were DNA, you'd have another side over here. I'm just going to draw quickly with a nitrogen and then a phosphate, right? So you'd have this side over here attached, which I'm going to show you on the next slide. All right, so a nucleotide is the monomer, and it's made up of these 3 parts, right? A base, a sugar, and a phosphate every time, right? So all 3 of those make up a nucleotide. So a pentose sugar is going to have carbons and oxygens in them. You can see here, just like most carbohydrates. And then here's where you get your phosphate group. So there's the P, the phosphate that's found in, the phosphorus that's found in every nucleic acid, right? So it's always got nitrogen, carbon, hydrogen, oxygen, and phosphorus, right, in every one. All right, so let's take a look at the 2 types of nitrogen bases. So I said some are 2 rings and some are 1. So we have, and you should remember this from when we go over DNA and biology, we have 4 bases that are in DNA. A is adenine, G is guanine, C is cytosine, T is thymine, all right? And so those are the 4 bases found in DNA. And the adenine and the guanine, those bases have 2 rings, so they're bigger, and those are called purines. So the purines are the bigger bases, all right? They've got 2 rings. And the pyrimidines are the smaller bases. They only have 1 ring. And again, the way that I remember them is the word pyrimidine has a Y in it, and so does the word cytosine and thymine. They both have a Y, whereas adenine and guanine don't. So we typically call adenine A and guanine G, cytosine C, and thymine T. Okay, and what we'll see is that across those, the 2 sides of DNA, adenine will always bond to thymine, and guanine always bonds to cytosine. All right, so a purine will bond to a pyrimidine and vice versa. Okay, so if I look at that, I'm going to come back here, but if I look at that structure, so here it is, phosphate, sugar, here's thymine, and across from it, I've got adenine, the nitrogen base, and then a sugar and a phosphate. And notice how they're flipped. They run opposite ways, and we'll learn about that when we get to DNA. It's not important now, but they are flipped, okay? So because of that, you'll get what's called a 5' end and a 3' end, but again, we'll get to that when we get to DNA, not important now. All right, so you can see that the pyrimidine bonds to a purine bonds to a pyrimidine, all right, and so on and so forth across there. All right, so I'm going to come back to that. And then there is another base that is a pyrimidine that's found only in RNA, and that is U, or uracil. So these two replace each other. So in DNA, you'll have thymine, and in all RNA, you'll have uracil instead of thymine. Okay, so that's a difference in the bases, and then we know that there's a difference in the sugars in the DNA and RNA, and we know there's a difference in the number of sides. All right, so RNA is only single-sided and DNA is double-sided. Okay, so those are the different nitrogen bases, the A, C, T, and G that you find in DNA, and then the uracil that you find in RNA. So RNA still has adenine and guanine and cytosine. It just replaces thymine with uracil, okay, in RNA. All right, so let's put them all together. So this is what it looks like. Again, here's your double sides. It looks like a ladder, and if I take it and I twist it, it becomes the double helix. All right, so notice that, like they usually say like the sides of the ladder, which is here, are my sugars and phosphates, and the rungs of my ladder are the bases that go across there. And notice that A always bonds to T, again, a purine and pyrimidine, and G always bonds to C, a purine and a pyrimidine. Okay, so you can see across here, C to G, A to T all the time. In between A and T, adenine and thymine, there is a hydrogen bond that connects them together down the middle of my ladder rungs. And there's only 2 hydrogen bonds in between the thymine and the adenine, but there's 3 hydrogen bonds in between guanine and cytosine. So actually these are held together a little more tightly than the A's and the T's are. All right, so it's harder to break between those 3 hydrogen bonds than it is between the 2 hydrogen bonds between adenine and thymine. All right, like I said, not super important right now, but I just want you to see how they line up. All right, so all of this, you can see just rungs of nucleotides, but then we've got a second side to it if it's DNA, right? So another side over here. All right, but again, it's just nucleotide, nucleotide, nucleotide. I'll circle the nucleotide, it's right here, right? A sugar, a phosphate, a base, right? And over here, a sugar, a phosphate, and a base, all right? They're just sort of flipped and they run opposite ways. All right, so that's my double helix. All right, I think that's it. Yep. All right, we're not going to go over the processes. So that's my little bit about nucleic acids. Okay. Yeah. So what I want you to do now is to finish your pattern matching and upload it. All right. So you're going to do that. We should be finished with it. So make sure you have everything done before you upload. So now's your time to finish if you haven't done the other slides. All right, so upload that to the assignment. I think it's called pattern matching. And then whatever time you have left after that, I would say just work on outlines because again, we'll probably get to a test by Tuesday or Wednesday, probably of next week. So we're getting down. We have a lab to do on Friday that I'll explain and we'll do that. We'll probably have Monday to finish up the lab if we need to, review, all right, and that kind of thing. Maybe give you more time to work on outlines. And then Tuesday, Wednesday probably would be the test. So Tuesday would be the multiple choice part, and Wednesday would be the free response questions, okay, with the retake then after that, okay? All right, that's your notes on nucleic acids. So go ahead and do the pattern matching and finish that up and upload.